I don't know if the seminars are more popular now. It's just Julia. The text for the attendance. When I see this, I think there's no need to change the time of the seminar, right? But we have some people that came and said it would be easier for us to come to the seminars in the morning, like Friday morning. It would be for coffee break before the seminar, and then the seminar. So we are going to do. There's a poll that is already
in the bulletin.
accessible in the bulletin. Yeah, there's some problems for you can leave comments. So please do vote because if we have only 10 people that vote for the coffee and no one else, you have to be significant. Okay, so please go to the bulletin and think about voting on your preference on the afternoon after a beer or in the morning, and we'll see how we could change on. Okay, and yeah, well, go ahead, please.
Thank you all for coming. No, okay. Well, most of you already know me from all the years I have been working here, but maybe you don't know exactly what I work with. So today I'm going to present a little part of my PhD, which I'm finishing this year. And the talk is titled Posidonia oceanica meadows as nursery habitat for fish. My supervisors are Jorge Terrados and Gilbert Hinn, and I'm in the MARES group, in the Marine Research in Ecological and Social Systems. So to start, I'm going to present the fish life cycle for most coastal fish. Maybe some of you are familiar with it, but maybe some of you are not. So the fish, when they hatch from the eggs, they are in the shape of larvae, which are usually pelagic, and larvae suffer metamorphosis after a short period of being larvae, usually between 5 days or 13 days. They suffer this metamorphosis, and they obtain the shape of the juveniles with the fins and scales. And juveniles already have the shape of adults, but are not sexually mature. And after a few months being juveniles, they mature their gonads and they become adults. The different processes that happen are called reproduction, settlement, when the larvae settle from the pelagic part of the column to the benthic part, and recruitment, when juveniles incorporate into the adult population. Today, I'm going to focus on the juvenile state, which is a very vulnerable state, and it's a transitional phase where all the processes that are involved can act as bottlenecks for recruitment into adult populations. So habitat selection is a very crucial step for juveniles in this phase. So this is where nursery habitats, why they are important. So let's explain what a nursery habitat is. First of all, it's important to know that you can find juveniles in many habitats, and not all of them are considered nurseries. This is because, well, according to the nursery role hypothesis proposed by Beck and collaborators in 2001, a nursery habitat supports a greater than average combination of increased density, survival, growth, and movement to adult habitats. For example, here in this graph, this could be an habitat where juveniles can be, but it's not a nursery, because if you see the size of the arrows is smaller here than here, which means that here density, growth, survival, and movement are bigger, but not all of them need to be bigger for us to consider a certain habitat as a nursery. It could be that only the density is bigger, or the survival, growth, a combination of two of these, or the four of them. Habitat selection, again, is important because For example, we can measure growth with the otoliths or density by visual observations, but some other, like survival or movement, are not so easy to study in the field. So habitat selection can be used as a proxy to study nursery habitats. Also, I want to explain what happens when juveniles move into the adult stage. Some of them move from one place to another, from one habitat to another. So in that case, the nursery habitat doesn't overlap with the adult habitat, but some of them overlap these two habitats. But the adult, of course, always moves greater distances, so the adult habitat is bigger. What characteristics make a certain habitat a good nursery habitat in comparison to others? Well, the optimal nursery habitats are found where there is an equilibrium between predation and feeding trade-offs. That means that fish found enough food and also shelter for predators. So the quality of nurseries is usually linked to a structural complexity or three-dimensional complexity of the habitat. Structurally complex habitats diminish predator efficiency by providing shelter and also provide greater food availability. Habitat selection is very important because individuals may select one habitat or the other, depending, for example, on the size of their cells. I mean, if the fish is bigger, they need bigger spaces for shelter than smaller. So some of the most important or common nursery habitats are mangrove forests, which have a complex system of roots where fish can feed and shelter, and also seagrass meadows, which I'm going to focus today. I think I don't need to explain what a seagrass meadow is, because all of you know Posidonia and that, but well, these meadows provide to the humans a lot of ecosystem services, which are benefits that we can obtain from these ecosystems. And some of them are coastal protection, carbon sequestration, recreational, and all the list that you can find here. But moving to Posidonia, which is the seagrass species I'm going to focus on, we see that it also provides a nursery function or service. This is because usually healthy Posidonia meadows have support a greater abundance and survival of juveniles than other habitats. But what happens with Posidonia? The current state of this plant is that the populations are decreasing because they are highly sensitive to direct and indirect human impacts, like boat anchoring, climate change, and this is leading to a significant loss of habitat structure and also changes in the ecosystem service provision. So, as a response to this concerning trend, the scientific community has been doing some restoration efforts in the last years to try to compensate all these structural loss that we are suffering. These actions have different objectives. One of them is to recover the seagrass per se, but they also try to recover the different functions and services that these ecosystems provide. For example, the return of fauna, and specifically the nursery function. So, as you see in the Mediterranean, the different countries have developed restoration programs. In the last years, they are very recent and at different scales. but we still don't know how they are recovering these services and functions. So the monitoring of all these actions is essential to know what's happening in these restored ecosystems. But for example, for the nursery functions, there are no previous studies on restored Posidonia meadows. In other seagrass species, there are not many studies, but we have found some studies studying adult fish recovery. For example, in Castro marina, which is another seagrass species, which grows faster than Posidonia. And in this work, they found that the fish recovery in the last years after a big restoration was like there was a recovery of the fish biomass. In another study, they found that between 2012 and 2018, the fish abundance and richness was also recovering in a restored Castro marina meadow. So restorations are providing benefits for fish, but we still don't know what's happening in Posidonia. So the objectives of my work were to assess the short-time recovery of seagrass structural complexity and the juvenile fish community within a restored Posidonia meadow in the northwestern Mediterranean in comparison to a reference meadow, which I'm going to explain in what we call chapter 1. And also we wanted to explore habitat preference of juvenile fish from two species that usually settle in Posidonia meadows, focusing in their active selection based on visual cues, which I will explain in chapter 2. For the first objective, we sampled in the north of Mallorca, in Pollença Bay, in a 2-hectare restoration project, which was funded by Red Eléctrica. It's called Bosque Marino. And these 2 hectares were planted in 4 sequential phases between March 2018 and February 2020. The colors here represent the different plantation phases. Each of these restoration units measures 50 per 50 meters. And inside each unit, there are 100 restoration nodes that are separated 5 meters one from the other. Inside each node, there are 16 fragments that were planted, making a total of almost 13,000 fragments. They all were collected on the beaches from Pollença Bay, and they were not they were not extracted from the substrate, and yeah, they were planted. After collecting them, they came from storms and other natural processes that were putting them on the beach. And apart from the restoration, we sampled in 2 control areas, which are the edge and the inner part of a reference meadow. And why 2 control areas? Well, there are several reasons. For example, some other works have found an edge effect, which is a concept that explains the differences found between the boundaries and the inner parts of ecosystems, which usually present more abundance and richness in the edge. For example, because there's a greater food availability. And also we decided to do 2 controls because we think that with our sampling method, there might be a bias in the inner part of the meadow due to complexity. I will explain this in more detail later. So once we had the 3 areas, we did the sampling. We sampled habitat structural complexity, shoot density, and leaf area index in situ in the field in 2 sampling seasons in the summers of the 21 and 22. And for the fish, we did diver operated stereo video transects, stereo dot, in seven sampling seasons during the summers of 2021, 22, and 23. Here are Maria and Chema doing the sampling. For the transects, we collected the videos on the field, but then we had to do a post-processing in the laboratory. We used stereo video because if we calibrate the cameras and synchronize them, we can measure the fish, which will allow us to separate the fish into juvenile or adults, and also to calculate biomass, for example. And it is a very precise method, more than visual censuses. For that, we use CG7 measure software, which is a specific software for identifying and counting fish. So once we had all the fish identified and counted, well, I'm going to explain the results now. For the habitat complexity, we obtained these graphs for the shoot density and leaf area index. In both of them, we see the same patterns. The complexity in the edge and the inner part of the meadow are similar, and in the restored area, it's still very low because Posidonia is a very slow growing plant. So as the restoration is not very old, the structural complexity is not very big yet. And we also saw that there are no annual variations. So we cannot say that the temporal scale we are using is enough to see some changes in the structure. However, we can say that the restoration was a success, as we found a survival of the fragments of more than 90%. For the fish, what we saw is that for both species richness, which is the number of species, or total density, we have greater values on the edge, followed by the inner part of the meadow, and followed by the restored. So based on this, we could say that the restored area is still not recovered in terms of nursery function. We wanted to see, because as you see here, there's a lot of temporal fluctuations that can be a result of many processes like seasonal patterns of fish, for example. So we wanted to see if there's a recovery over time or not. So we plotted the differences between the restored and the edge in blue, and the restored and the meadow in orange. And what we expected if the function was recovered would be like this, like it goes recovering until it reaches 0, which means that there's no difference between the edge and the restored, or the meadow and the restored. But what we saw here is that there are no significant differences. So even though we can see it, that the slope is slightly going to the recovery state, we cannot assure that there's a recovery over time for the moment. And in terms of community, we wanted to see which species were using the restored area. And we saw that in the edge and the inner part of the meadow, there were many species, but on the restored area, they were mainly 2 species. These 2 species were Diplodus annularis, which is a sparid, and Symphodus tinca, which is a labrid. And we didn't know if these variations, because as you can see, these 2 species are also present on the other habitats, but in greater abundances. Well, this is global dominance, which is like the contribution to the whole community. But you see here, the values are lower. So we wanted to see if these variations were caused, for example, by differences in mortality or survival, because, for example, here the complexity is smaller, so they would be more vulnerable, or also if they were selecting more these 2 habitats instead of this one. So we proposed the second chapter or objective, which was a laboratory experiment to explore the habitat choice of these 2 species, in focusing on visual selection. We went to the field and tried to collect them. We used different methods. For Symphodus tinca, it was easy. We used this trolling net called gangle, and they were easily collected. But for Diplodus annularis, we had some problems, and we had to try different methods. So we finally did it with hand nets last summer. When we had all the fish we needed, we went to Linia, to a laboratory that it's located in Portland, and we started doing the experiments. Here is the aquarium we used. It's a square aquarium with 4 corners separated from the central arena. And what we did was presenting 4 different habitats in the 4 corners. One of them was a control. Here you cannot see it very well, but there's no habitat, no Posidonia. And the other 3 are Posidonia at different complexity levels. We put the fish one by one on the center of the arena and recorded them for 15 minutes. The first 5 minutes were not considered because the fish were acclimating to the aquarium, but then we counted how much time they spent near each habitat. For doing that, we used a free software called ID Tracker, which, as you can see, detects the fish in red. And after the 10 minutes of recording, we obtain these kind of tracks so that we can say how many frames or how much time each fish spent near each habitat. For that, what we did was putting a grid over these squares and classify each small square into an habitat. The blue one that you see here, it was considered like undetermined, like the fish was not selecting any of the corners, and the other ones were considered like the fish was selecting the habitat that was in that corner. So for the experiments, we separated them in 3 sets. In the first one, we manipulated the density. We used long shoots of artificial Posidonia, and we used 3 levels of density. For the second set of experiments, we did the same, but with shorter shoots. And for the third set of experiments, we used the lower density used on the other ones, 200 shoots per square meter at 3 different heights. For that, we used 30 fish of each species in each one of the 3 sets. So, and that made a total of 180 fish. And complexity, I told you that we measure it as height and density, but we also wanted to study the 3 experiments together. So we wanted to have like a complexity measure that could compare all of them. So we decided to use surface area and fractal dimension. The results in terms of complexity were that when the complexity was lowering, also when the density was lowering, the complexity both in surface area and fractal were also lowering. The same was observed in the second experiment, and the same in the third with the height. So we can say that surface area and fractal dimensions are good indicators, or they reflect good changes in height and density. So they are good indicators of complexity. In terms of fish response to the experiments, we saw differences between the 2 species. Diplodus annularis is a species that only recruits and lives in Posidonia, while Symphodus tinca can recruit also in macroalgal forest and other habitats. So what we found here was more or less what we expected. Diplodus annularis always saw a preference for complex habitats, which are this one, and this one. And the pattern was always like from more to less complex. While in Symphodus tinca, which is more generalist, we didn't find any clear patterns. What we found in the two species was that they prefer the three Posidonia habitats over the undetermined area in the center and the control corner. So they were able to differentiate the habitats and select them. When we evaluated the three experiments together in fractal dimension and surface area, we standardized the complexity of each experiment to be able to compare them, and we saw the same, more or less, that Diplodus annularis presented a significant preference for complex habitats, which would be this one. And Symphodus tinca, however, didn't present any preference. The line has almost zero slope. So then if we go back to chapter 1, we can see what we wanted to see what was happening over time. Like we would expect that Symphodus tinca would be recovering faster in the nursery function because they are not so so specific with the habitat conditions than Diplodus annularis. So we plotted this same again for the two species, and we saw that, for example, Symphodus tinca is, if we compare it to the meadow control, we could say that it's almost recovered. The line is on zero all the time. And if we compare it to the edge, it's also going to the zero. So that's what we were expecting, that Symphodus tinca is recovering faster than Diplodus annularis, but still we need to do this kind of studies in the future, because the temporal scale that we are using is still not enough to know what will happen at the end. So to finish, I'm going to read a few highlights of the work. The structural complexity in the restored meadow is still far from reaching values comparable to the reference meadow. Some fish are starting to use this area, but the recovery of the nursery function is still in its early stages. Juveniles of Diplodus annularis and Symphodus tinca are capable of differentiate and select Posidonia over unvegetated areas, and habitat selection patterns are species specific. While Diplodus annularis are more dependent on complexity, Symphodus tinca are not dependent at all. The recovery of the nursery function is expected to happen faster for Symphodus tinca than Diplodus annularis. And that's all. Thank you all for coming, and I'll leave you with a video of annularis.
Do you think there would be any value in trying to do a sort of joint restoration of the plants and the fish? Meaning that you provide also at a certain point the equivalent of what in microbiology would be an inoculum, right? Like a small population of different species that reproduces, in some sense, what you would find naturally in a well-established Posidonia meadow.
I don't know how you could do that. I mean, the habitats are close, so they are supposed to be able to move between one another. But I think if you put them directly, they maybe will move back to the other habitat. I don't know, but
I didn't know that they were.
yeah, they are like 100 meters from us. Yeah, that's why we expect to recover, because the fish can go from the donor meadow to the restored meadow easily, right? So
I really like a lot the work. Congratulations. I have a couple of questions. How do you know when you do these transects with the video cameras that you are not counting the same fishes all the time?
we try to separate them as much as possible, but I mean, maybe there are some fish that are
Yeah.
related. Yeah, that's
Yeah, this is a problem also that we have when we see
Yeah.
pollinators around, that you never know if you are counting them again and again.
yeah.
And I guess there might be some differences between species because some of them might move more on the same, might be more territorial than others. Right?
Yeah, right. Yeah.
And about this, can you put the graph with the species of fish that you saw in the restoration on the other?
This one?
Yeah, this. You said this is percentage of dominance?
This is maybe dominance is not the one. This is the percentage that they contributed to the total. Like
So I don't understand why in the restored area, if you just have two fishes, why this is not in percentage total? I mean, why it's not 50 and 50 or 40 and 60?
because they are and it's calculated to the total like
Ah,
to the
okay.
overall community on the three habitats. Yeah,
Okay.
there's
Yeah,
also
I
have it
understood it
calculated.
was in each.
No, it's for the three of them together. I also have them like for each habitat independently, and it's still the two same species, but yeah, they some 100.
And then I wonder if you see this is like you in the normal nice one, you have many fishes, and then in the restored, you just have two of them. And then you did the experiments with the two of them that are everywhere. So they might not be that selective as the others that are not in the restored area. So I wonder why you decided to do the experiment with these two that you have in the restored area and not in the other areas, because I guess those that you have in the normal meadows
Yeah.
and not in the restore, maybe they don't like the restore because it's not
Yeah, it's not
complex
enough.
enough, or, so why would you decide?
Well, there were several reasons. One of them is because they are two of the more abundant species, and also we have to collect them and kill them. And so if we take some species that are not so abundant, it's not so easy to collect, and also you are maybe hurting the population more. And also because I would assume, for example, Diplodus burleis, this green bar here, that, okay, seeing this, I already know that they will select only the more dense. So we wanted to see if these two were here by chance or they were selected, but with this one, you can assume like that they really have a selection pattern. But yeah, it's true that we could have done it with any of these species.
And do you have data that are not experimental, but like relative data on the complexity and size of different meadows related to the different species that live in there and everything? Or this is not that easy to have?
I don't have this data.
I
Yeah.
mean, I don't know your group, or
Yeah, I don't know. No, I don't think.
I don't know how easy is that. Because then it could be cool to check
Yeah.
how differences in the meadows are having an effect on the fish community, because there are many interactions in there.
Yeah. But I don't know if that data exists for human eyes because they are very understudied. So yeah.
And you said you kill them?
Or yeah. That's
Very well.
that's mandatory for
experiment.
ethical. Yeah, I don't even know the name.
Ah,
Yeah.
after the experiment.
Yeah, the experiment, yeah, the national takes off. Like if you do animal experiment, you need to kill them
And what's the reason?
because you can be transmitting them some diseases or so if you release them in the ecosystem, you can be spreading something that for us is not harming, but for them,
Okay.
yeah.
Thank you.
So actually, I wanted to go back a sec to what Amparo was saying before, in regards to what goes in a sense below in the trophic level from fish. Do you know, is there any study at all on things that are smaller that could be, for example, the things that the larvae eat? Like, I guess it's gonna be, I don't know, copepods or things like that. Is there any study of the microbial population in nursery, sorry, in what you call the restored areas versus the well-established areas?
Not that I'm aware. I don't know about any.
Yes, there are studies in other restoration projects, and also we are also sampling the epifauna, small crustaceans that live in the meadow, which would be the food for some of these species. We are also sampling them to check if they are recovering also. We expected that they would even be faster in recovering because their life cycles are shorter. And what we have seen is that still recovery is not much compared to the meadow. Still, there is less abundance of all those epifauna, which are mostly copepods, mostly crustaceans, which are one of the dominant groups.
In Prostera, they are recovering faster, I think, but in Posidonia, I don't know. And about the microbial community, I don't know.
Yeah, no idea.
It's a bit early.
Yeah, this is like a preliminary in 20 years, maybe. We can start.
I have a maybe stupid question, but that's a very local study, but when you talk about fish biomass, how do you assess at higher scale? Like, maybe not in Western Mediterranean, you said before the fish biomass in the Atlantic is recovering.
Yeah, here I would say only at a local scale, but you can measure biomass from the length of the fish. There are some coefficients for each species. And if you apply a certain formula to each fish based on the abundance and the size, you can obtain like biomass for each individual or community. But it would be very local here. But
No, it's all from sampling.
Yeah.
You go there and you just.
yeah, you have an area, you sample an area that you know, so you know how many fish are in a specific area, and yeah.
But if I want to know the whole Western Mediterranean, you said yeah, Atlantic. So how do you how can you extrapolate to the whole?
No, but here they
No.
in the Atlantic they did a study in a certain part of in the coastal lagoons where they restored the meadow. They did not
In
sample
smaller
them. Yeah,
smaller
it was also like local,
Okay.
bigger than ours, but it was also local. Yeah, maybe in fisheries there are ways to calculate biomass at a bigger scale, but I'm not very familiar with this.
Actually, I have a curiosity question, which is not directly related, it's more related to restoration of Posidonia. So if you say it's a restored area, means there was Posidonia before and then it's gone because of environmental factors.
Yeah.
So what did you do then to the plants that you restored, like the new fragments? Are they anyhow differently implemented so that you can be sure, or you try to ensure that this meadow will survive? Because otherwise, if the environmental factors are just getting worse, like say heat of water, Mediterranean, more acidity, whatever, you cannot. I don't know what makes you believe that this meadow will survive or will flourish.
First of all, it's protected by boats, so boats cannot anchor in there. So this is one of the main problems for Posidonia. And about temperature and acidification, we cannot do anything. But also the fragments are planted with with a metal thing that puts them on the substrate, and in the first stages they are cold to the substrate very hard, so they are. It's not easy that they go away or anything. For example, with the storms, but yeah, I think the main thing that we can do is trying for boats not to anchor them there.
We actually, in the context of climate change, you can do anything.
Mm,
And restoration.
no, no. I think you can only control anthropic factors that, and some of them are not possible to control.
Just for curiosity, one question. Let's think that the climate change will not kill the restore area. How many years do you think that will need to get the same abundant fisheries than the meadow that we are comparing? 25 or 100 years or 50 years?
I cannot say a number, but maybe 25, 50. It depends on how the Posidonia, like, for now, the growth is small, but they are growing, but maybe at some point they will stop growing or not, so I cannot really say, but I would say that at least 25 or 50 years,
Okay.
or even more.
Are you planning, not you? Are you planning in general to follow this for many years for the country? I think it would be very cool if you could track also interactions in different trophic levels, and because in I don't know if you know, in Seychelles there were these restoration projects and that remove invasive species, and then they check how the interaction networks in different levels were, whether they were restoring. So not only the species, but also all the processes through the interactions. So if you could manage to do that, that would be really cool.
One of the main aims of the project with this restoration is to establish experimentally, precisely to start doing all these kind of studies of experiment, having all the media knowledge from the beginning, the history, and the thing is that probably new people coming to do this experiment, and also we have to be sure something that we also mentioned before, that we can maintain the area. Not only from climate change, which is something that we cannot control, we have increasing temperature and we have mortality, but also from the local disturbances that are very important. Posidonia living in a particular place. And this project is a joint effort with the government of the Balearic Islands, and the government is, in theory, will be the responsible of maintaining this area in the future. Let's see. Let's see.
And why did you plant it Posidonia in different years?
I was not here before.
But is there a reason?
Yeah, I think so.
Because of availability of resources in time. And the season, not all seasons are the same. For example, summer is not a good time.
And have you seen differences in the?
No, they all follow the same track, no? With some months in the day, but they are behaving more or less.
That's better.
Yeah, that's why I consider everything like
Yeah.
continuing.
I didn't know if it was part of the design that you wanted to include variability.
No, it was for practical.
Very nice.
Did you think about using a different species of plant, like monotheria or a faster one? I don't know. Because Posidonia is super slow, maybe a species.
Yeah, but I mean, in that area, there was Posidonia before, so I guess that the main objective is to recover what it was there before. So probably you can restore Timothea, but depends on what you are trying to do. So here, I think it makes more sense to try to grow Posidonia because it's the species that was originally there. So that.
There is about there.
Yeah, there's Timothea in the area.
Has colonized.
Yeah, but maybe in some other areas where Timothea was there and it's lost, it's it's good to plant Timothea also. So, yeah.
And that's actually a very nice subsidiary question. In places where it was lost, Posidonia, and assuming that is going to take a huge effort, economic and even a long-term effort, does it make sense to, for example, to restore with other plant, a different plant, or?
For me, if you see it from the, for example, ecosystem services perspective, Timothea is not the same for fish, for example, than Posidonia, because they cannot shelter and everything the same. So maybe in terms of planting some other species or not planting, it's cheaper. But if you want to restore some of these ecosystem services, you need to restore Posidonia and not other species. Because they act differently and they have different functions and
but you have a balance there. Either planting something that you actually will see in 25, 30 years, or something that at least can give you a certain services, for example, to diminish sediment water suspension, and it's a shorter term fluctuation.
I mean, I see the advantages of that, but I also see the advantages on the other. So I think it's the same. It depends on what if you just want to stop sediment suspension quickly, of course.
And I think just for example, in terrestrial ecosystems, where, for example, there were oaks at the time that they are very slow growing species, and just to avoid land loss or whatever, they use other species much faster growing, etc. So there's a balance between what you can actually achieve and the terms on which that is achievable actually.
So the single fish experiments that you have done, right? If I remember well, there was at least one of the species that were opting for the corners that were visually more complex. Was visually more complex, right? Because they were separated. So I was wondering whether it might be interesting or useful and not useful to sort of plant artificial structure that create a higher complexity locally, whilst the thing is the natural bit is recovering, in order to attract fish to live there.
There are so many, and there are many experiments and actions doing that, planting in artificial reefs, or so at least they are creating complexity for fish or invertebrates to go there.
And it works.
It works. It seems to be working.
The first experimental studies that were looking at the association between fish and seagrass. We're using artificial plastic seagrasses, changing densities, changing size of the flow, changing height, to look at what would be the effect of how the fish will respond to that. So that would be a classical approach.
And do you know if any of these experiments have, linking to the previous comment, have ever tried to first recover some fast growing Histosera, for example, to then facilitate Posidonia? If the sediment stabilizes with some other fast growing.
I don't know if that has been done, but for example, in our area, there's Timothea there. So, I think Timothea facilitates Posidonia settlement. So probably there are some experiments on that, but here we had it naturally. So there was no need to plant a faster growing species before. But that could be another approach that could be useful.
I think that there is a question on the chat. So, from Julia Maya.
¿Y cómo se hace esto?
Bueno.
Ya he dicho, explícalo en chat.
Ah, a ver, es que está muy lejos, ¿no? No sé.
Yo tampoco. Sí. Hola. Hola.
No sé. No, I can text her later. She's not.
Ah, but
Maybe she left.
it's in the, she put it in the YouTube.
Ah, okay.
Oof. Well, maybe, sorry, Julia, maybe.
I have the question.
Okay.
So, is there less variability in species richness between seasons in the restore meadow compared to the other two?
Variability in, sorry, can you?
Yeah, related with the chapter 1,
Yeah.
as far as I understand,
Yeah.
like if there less variability in species richness between seasons in the restore meadow compared to the other two.
Yeah. Yeah, here you can see that as richness is always smaller here, except for May 2023, it's more or less stable, while here you have more variability. In this four, it's similar, but yeah, I don't know what happened in May
And if you
2023.
statistically measure it?
Yeah, and it's significant. There are more different. Yeah.
That was the question.
Vale, pues gracias, Julia.